So this is the very high level definition of the approach we use for signal processing. First, we referenced the data with actually we had the reference electrodes, but we did another referencing with the average of T7 and T8 in the temporal zone electrode. Then we did the low pass filtering because we didn't want the high frequency noises to have this to be included in our images. And then we did the downsampling because our biosemi device has a sampling rate of 248 hertz. So we downsampled it to 256. And then we used the high pass and notch filtering. High pass filtering was basically from 1 hertz to 128 hertz. The cutoff frequency was only 1 hertz. And this was done because to get rid of the very low frequencies. And notch filtering, of course, because we didn't want the city line noise to be on our system. And then after this, we used the discrete wavelet transform to decompose the signal into different bands. As you can see here, we used WG6 wavelet, which was proven to be good wavelets for the analysis of EEG signals. And we decompose the signal into different bands. So as you can see, the detailed and approximation bands that will be provided by wavelet decomposition shows the range of the frequency of the signal. And at the end, the A6, D6, D5, and D4 bands were summed together to reproduce to build the P300 signal. Here are the results of these approaches for the first experiment. As you can see here, this is the result summed over all the subjects in experiment 1. This is the average forms. As you can see, for P3, PZ, PO3, PO4, P4, and CZ electrodes. As expected, the P3 had the largest amplitude here. So this will show the degradation of amplitude of P300 as we reduced the time T1 and T2 times that I described before. So in the first image, as you can see, we used 500 milliseconds for T1 and T2. So that means one image per second. And as you can see, we have the largest amplitude of P300. And as the T1 and T2 decreases, in the worst case, at the last experiment where the T1 and T2 were 50 milliseconds, so we had 10 images in 1 second. As you can see, there is still a benchmark of P300, but the amplitude is different. Another observation is that after the third experiment, which was the image rating, image rate, presentation rate was 3.3 hertz. We can see another type of benchmarks in the signals, which now we know that is called steady state visual evoked potentials, which are exactly the same frequency of the image presentation rate. And the result that we obtained based on this signal can be also partially due to the difference of this SSVEP of the two classes. This is the average signal of the third experiment when we had 3 classes and we had subjects, expert and non-expert subjects. So as you can see, this image is for the subject provided by ACT, and all other figures show the P300 of the other subjects. The red line is the P300 correlated with the target images, obvious target. The dashed line is P300 correlated with non-obvious targets. That's that little spot that you saw in the image database description. And the blue one is correlated with the non-target ones. So as you can see, there's a benchmark, as expected in the previous experiment, P300. For the expert, actually he didn't react upon the non-obvious targets because he didn't consider them as targets. So as you can see, there's no P300 for the non-obvious targets, but for all other subjects there is something. which the shapes and the latencies and their amplitudes are different somehow. Sometimes it's even bigger, but there is a benchmark. But for the expert subject, there was no. These are this is the F measure of the classification system using, actually I forgot to talk about when I talked about signal processing algorithms. As for classifications, classification of signal, the final extracted P300 signal was again decimated to 32 hertz because the maximum frequency component of the signal was 16. This decimation adds another low-pass filtering. That the cutoff frequency was 12.8. So at the end, the P300 signal had the maximum frequency of 12.8. And then these 32 samples of each single trial of different electrodes were concatenated together to create a Fisher vector of each single trial. We used 16 electrodes. Actually, we have 32 electrodes, but we selected only 16 electrodes in basically from central to occipital part. And then we did a PCA to reduce the dimension of the data to 288. And then we used 5-fold cross-validation using SVM. And this is the F measure results obtained for different subjects. So as you can see, the x-axis shows the different experiments that we have, the reliability versus speed. And the y-axis shows the F measure obtained. So as you can see, for the mean value of F measure for experiment 1 can be 70, and it goes down for the last experiment. For some subjects, it's even below the random guess, but for the 2 best subjects, it's higher. And in the second experiment, here are the results obtained for the different classes, and here you can still see that for some subjects in the second experiment, the measure of 70% can be obtained. So the conclusions, we studied in this project the possibility of selecting scientifically interesting images using EEG signals. To these and different interstimulus intervals values were explored. And it was seen that as the image presentation rate increases, the performance degrades. Also, we saw that there is a clear difference between P300 correlated with seeing obvious and non-obvious targets for the expert subject. And the other observation was this P300 pattern is different among subjects, so it's really challenging to build a general classifier that can be used for everybody. Thanks for your attention.